Thank you, sir. So first of all, I thank you for accepting our invitation to give a talk in AppCare IEEE AI Symposium. So let me introduce about our speaker, Professor Ashwin Srinivasan. Professor Ashwin Srinivasan is the class of 1981 chair professor at BITS Planning, Goa. He received his PhD in 1991 from the School of Electrical Engineering and Computer Science in the University of New South Wales. Professor Ashwin joined the ILP group at the Turning Institute, Scotland, and with Stephen, now at Imperial College London, worked on the development and application of inductive logic programming. From 1993 to 2003, Ashwin was at the Computing Laboratory as a postdoctoral researcher in Oxford University. Then the Nuffield Trust Research Fellow in Medical Mathematics, and finally as a university lecturer in computation. At Oxford, he was involved in pioneering applications of ILP systems to difficult real-world problems in molecular biology and chemistry. In 2003, he moved to the IBM Research India as a research staff member. In 2009, he was awarded a Ramanujan Fellowship by the Department of Science and Technology of the Government of India. In 2010, he took up the post of professor at the newly formed South Asian University and became the founder dean of the Faculty of Mathematics and Computer Science. From 2012, he was professor of computer science at IIIT Delhi and moved to BITS Planning, Goa as chair professor in January 2015. From 2020 to 2023, he was the head of AppCare at BITS Planning. Sir, you can start your presentation from now. And for participants, if you have any questions, you can raise the hand by clicking of the button below and we will unmute you. Also, you have another option as a chat. So you can, if you have any mic issue, you can just chat, type your questions on the chat. Thank you, sir.
Okay, thank you, Srinivas. I don't know exactly how many of you are present, but thank you for attending the talk. A few corrections. I'm no longer the class of 1981 chair professor that ended earlier this year. And I actually finished my PhD in 1991, which doesn't make me, it just makes me marginally younger than finishing your PhD in 1981. But anyway, I suppose when the numbers are so large, who's counting? So today I'm going to tell you a little bit about some recent work that I've been involved with my student Shreyas Bhatt and my former PhD student and now postdoc at Cambridge, Sitraj Dash, and some of my colleagues from TCS Innovation Labs, Lavkesh and TCS Life Sciences, which is Arijit Roy and Saumya, and also my colleague at BITS Pilani Life Sciences, Biological Sciences, Ravi Duluri. So quite a lot of people all have contributed in interesting and useful ways to this talk, which is on drug discovery using large language models with logical feedback. So let's get started. Please interrupt as and when you want, because I'm happy to take the questions as they come. In this talk, I'll give you a little bit of a background of computer-aided drug design, which is sometimes abbreviated as CADD. I'll then talk about generating molecules using large language models with logical feedback. What does that mean? Show you some results from early experiments that we have. I'll talk about future work and the bigger picture involved in what I'm presenting today. So almost the whole story is in this picture. Now I recognize that most of you are probably too young to have even seen a hand pump, but you may have heard of the existence of such a device. It's a pump for water, quite often seen in India before, but certainly probably still seen in rural India. So the pump basically takes in some primer, which is you have to pour in some water to get it started, and then you press the handle up and down, and the pump draws water, sucks it up from the ground, and then sends it out through its output. Now, you can think of what I'm talking about, the hand pump, as being a large language model. It's slightly more sophisticated than a hand pump, because it's basically going to generate water or molecules. For us, the water output is similar to molecules. But the large language models are slightly more sophisticated than a hand pump, which is good, because if all we did was to use a hand pump, then that would be a bit non-productive, I guess. But so they are different in the sense that the output of these models tends to be more and more like the input as you feed in more stuff back. So if you're giving it some primer, then it tries to generate sort of colored water like the color of the primer. So assume this is what's happening, that the pump is generating some molecules. They go to a filter, which is that constraints on molecules cloud that you see, and that filter basically separates the molecules generated by the pump into some bad molecules, red, and some good molecules, green. The good molecules initially are forced to flow back into the pump, replacing the original primer. And because this is not just a hand pump and it's a large language model, that feedback that it gets, it sort of uses that to generate more greenish molecules. So you can think of it as changing the distribution of the space of all possible molecules, rather than drawing water from the space of all possible molecules. It will be drawing from the space of sort of greener and greener molecules. So as you keep pressing the pump, more and more stuff comes through, more red goes into the bottom, more green gets fed back. Finally, at some point, the feedback is turned off through a valve, let's say, on the top. And then what happens is that you get the output of the pump going through the filter, the good ones going not back into the pump itself, but going into the reservoir on top, which is a bunch of good molecules. And the chemist has a look at those. So that's basically the full story. So the logical feedback, everything is sort of roughly happening like this through the constraints, which are constraints in logic, and the feedback is essentially fed back to the large language model. Let me come to the problem at hand, which is drug design, which is I have characterized here based on Thomas Hobbes' famous line. So drug design notoriously is difficult, it's wasteful, it's costly, it's uncertain, and it's a long process. Now, let's have a look at some of the main tasks in this long process. Let me zoom this in a little bit. So it starts roughly with the idea of a target. So what kind of molecule are you trying to interfere or block or do something to? Usually a protein. So that protein has some part of it which is chosen as a target, and you get some structural model. Structural in the sense that where what is in three-dimensional space of that part of the target that you want to interfere with in some way. And based on this model that you have, it's a model because you may not have the real thing. You may not have a crystallography structure of that. So you may have a model of it, say using something, I don't know, like AlphaFold or something like that. You get what are called ligands. Ligands are small molecules that somehow lock into this site or this active region of the larger molecule. There are a number of computational tasks that go into each of these sub-activities, and I've outlined some of those here in below the dotted line. I won't go into what they are unless people want to know. But so yes, we get these small molecules, ligands. We try to predict how well they will be. Once we have a reasonable prediction, suppose they're not so good, then you go back, get some more ligands, and you keep doing this. And once you're reasonably happy with what you have, then a chemist tries to synthesize these compounds. That in itself is not obvious how you would do it in some cases. So once a chemist has synthesized the compounds, then they undergo some biological assays and trials. Once they may not work out so well, in which case you come back, change the model of your active site, restart the whole cycle again. Finally, when everything sort of looks good after the biological trials, then they go on to safety trials in animals, mice perhaps, and then it goes on to human several phase trials. So the whole process is involves several activities and may not actually be as as immediately obvious at each stage as to what is going to happen. So I told you that we start basically with the notion of a ligand, and the protein is, say, shown here as a sort of as a lock. So this is a famous analogy or well-known analogy where the target site that you're looking at is on the protein. So you're trying to find a small molecule, this ligand, that fits into parts of that target site. So the ligand is like a key, the protein is like a lock, and what we're trying to find is keys to locks as such. Okay, this is going the wrong way. There are, so just to make sure that we're talking about the same sort of things. So what I'll call ligands are molecules that can bind or fit into the lock. But that's not enough. So there are additional constraints that chemists impose on ligands, and molecules that satisfy those constraints are called leads. Drugs are much further down the line. They've been shown to satisfy their leads, which have been shown to have some therapeutic value, and additional sort of legal constraints like they should be patentable and so on. So ligands, leads, drugs. So ligands are just the small molecules. Leads are small molecules that satisfy some constraints, and drugs are leads which satisfy some additional constraints beyond the chemical sort of constraints. Now, usually potential ligands are obtained by screening libraries of known compounds, and then modifying them. Now, the set of known compounds is of the order of some millions. But the problem is that the space of possible chemicals that you can build is of the order of decillions, like 10 to the power 60, as opposed to 10 to the power 6. So using this access to this database of about 10 to the power 6, we want to hypothesize new ligands and leads from this space of 10 to the power 60. So obviously we have to be somewhat creative to do that. And those of you who have might have read, say, well, it's not important, but one possible way to be somewhat creative is to hallucinate. So what we need is really some kind of sane hallucinator. And this obviously is the key hook into which I am going to introduce the use of large language models. Because while large language models, the hallucination feature of large language models is usually presented in a negative aspect, it's actually quite a good thing for us. We want things that can think out of the box or generate ideas out of the box, but they have to be somewhat sane, and that's how we come to this notion of logical feedback, which will introduce the somewhat sane aspect. So, so the basic process that I told you, this is just a block diagram version of the molecular pump story, that the hand pump story that I told you. And this is actually what's happening in the programs that we use. So the constraints, chemical constraints that are imposed on leads, along with what we already know, initial background knowledge, and a query and some examples, all come in and are assembled into a prompt. I'm just going to work through an iterative process that happens. So a prompt is constructed using the constraints that we want to satisfy, what we know already in terms of the molecules that we would, the properties of molecules that we would like, and so on, and some initial data, if we have it, that we know that some of these molecules are known to inhibit the target. So stuff from previous experiments, data, so on. And a query, which we will see, is very simple. I'll show you what each of these are in a moment. And we construct a prompt. So out of this refined prompt comes a prompt, which goes into an LLM. The prompt allows the LLM to generate examples, which are molecules, which are then checked with the constraint, with an external constraint checker, which happens to be just a logical inference engine in this case, because the constraints are all logical constraints on things like, molecular weight should be between this and that, and then affinity should be at least this much, and so on. And so the molecules, not all of the ones generated by the LLM may satisfy those constraints. So we will come up with some result of this constraint checker, which will give us some new examples, which are classified, say, into good and bad, those which actually satisfied the constraints and those that didn't. So these new examples are one thing that goes back into the refined prompt. Remember, the refined prompt takes constraints, it takes what we already know, some initial examples, and a query. So some new examples now go back to the prompt constructor. The background knowledge is updated because we now know that there are some new molecules that do and don't satisfy the constraints. The constraints are refined, and this is an unusual aspect of it, of what we do, is that we also not only update or refine our background knowledge, and we not only update the examples that we have, we also update the constraints in a particular way so that it becomes more well, restrictive in one way and more generalized in another way, which I can explain in a moment. But so all this new information goes into the, constructing the prompt. So we get new constraints, new background knowledge, and then new examples, and they all go back into new prompt, which is constructing a new prompt, which is then again goes back to the LLM, again generates molecules, the whole thing is repeated. Okay, so that's roughly what is going on. It's not very different to the molecule pump story that I told you, but there are some questions that follow naturally. So what is the role of the LLM, which is obvious enough, hopefully, but I'll nevertheless say something on it. So what are in the B's, C's, and the P's? P's are the prompts. So what is in the background knowledge, the constraints and the prompts? And what's the role of the query over here? What is that query doing? And how do we change the background knowledge, constraints, and the prompts? And why do we bother with the iteration? So here are sort of 5 questions which I will talk a little bit more about. So what's the role of the LLM? So on each iteration, the LLM generates new molecules. It's basically just doing an auto-completion of something which says, you know, can you complete a good molecule, when it should tell you, okay, here is a good molecule with some instantiation of molecule, conditional on the prompt that it's provided. So we're not training the LLM or anything, it's just conditioning it. But it's just generating hopefully good molecules. So B, the background knowledge, denotes what we already know. For us, it is a logical theory. It includes functions for evaluating the constraints, facts about the molecules that we have generated so far, and really it should also, you could easily include C, which is the constraints, but I've chosen to separate them just to emphasize the fact that some part of what we already know are constraints, and the fact that this constraint is there is important to realize. So C denotes for us chemical constraints that are satisfied by molecules. So it's you could think of it in logical terms as a unary predicate. So C of X, where X is a molecule, is either true or false. So on any iteration, there are a set of constraints. P contains the context in the form of some instances that we already know about, the molecules that we have generated, which have been now labeled good or bad, depending on whether the constraint predicate evaluated to true or false. This is essentially the logical feedback that happens. Once the constraint is evaluated to true or false, it is then fed back into few short examples or instances into the prompt. The prompt and all that is automatically assembled, of course. So nothing much is there in the prompt other than just this context. And the query is a very simple query. It just says, can you generate good molecules that are not in any known database of chemicals? And that's the interesting part of it, I suppose, if at all, is that we would prefer to see molecules that we haven't seen before in any known database. Doesn't mean, of course, that they may not be in some proprietary database. Whatever the language model knows, it should try to generate molecules which are not in that in what it already knows. So between them, the query and the prompt are all very straightforward, and this is an important aspect of what we were trying to do, because too much of existing LLM experiments depends on the cleverness of the prompt. So we wanted a pretty straightforward prompt as such, so that we could repeat the experiments without, so we didn't want any human intervention in generating the prompts. So it wasn't dependent on what, say, a chemist knew or somebody else knew about how to write prompts. So prompts are very straightforward, automatic conversions of what is already known with this simple query, which is the same on every iteration. This doesn't change. We do, however, try to ensure some kind of weird bracketing problem here, that when we update the background knowledge and the constraints, we are trying to satisfy a kind of monotonic property of the new background knowledge entailing the previous background knowledge, and the new constraints entailing the previous constraints. For those of you who haven't seen this logical entailment symbol before, all it's saying is that whenever the left-hand side is true, the right-hand side must be true. So if you take the constraint, any molecule X that satisfies the new constraint will automatically satisfy the previous constraint. But it doesn't mean that anything that satisfied the previous constraint will automatically satisfy the new constraint, because then there would be no point in changing the constraint. And we'll see an example of what I mean by changing the constraint in a moment. So the prompts are refined just simply by slot filling a template using the current values of the background and constraints. So why this iterative procedure? So this iterative procedures, for some of you who have long enough memories, is actually inspired by an algorithm called MIMIC. MIMIC progressively samples, it's a density estimation algorithm, which basically samples from a distribution in which optimal and near optimal solutions have increasing probability on each iteration. But it does this by retraining the density estimator, usually using some kind of machine learning. I mean, one way to do it is to use something like a machine learning model to do the density estimation. The MIMIC's real utility is in cases where optimal solutions are fairly rare. So if you just randomly brew from an unconditional distribution, then the chances of you getting an optimal or a near optimal solution is quite low. So the reason what you're using this iterative procedure is progressively skewing the distribution so that you get optimal solutions or near optimal solutions having a higher probability. So we're doing somewhat the same thing. We're not learning a new density estimator, but we're progressively sampling from different conditional distributions by changing the context encoded by the prompt string. So each time we update the prompt string, we're changing the context, we're changing the conditional distribution that we're sampling, the LLM is sampling from. For the present, we can assume that the LLM is not the only thing that we do, but we can assume that the LLM is used to generate molecules, ligands, with increasingly higher binding affinity to the target. In fact, there are a whole bunch of other constraints as well, like on molecular weight and synthesis accessibility scores and things. I won't talk about that, but this is an interesting one to tell you about. So, for example, on iteration I, we may want the LLM to generate molecules for a target which has an affinity less than or equal to -8. So most of these affinity procedures generate negative numbers. So the more negative than the number, the higher the binding affinity as such. So a molecule that has -10 or below is better than one that has -8 or below, in the sense that it's estimated to bind stronger to the target. So on iteration I, let's say we make sure that we generate molecules with affinity less than or equal to -8, and then on the next iteration, you remember I said that we refine the constraint. Well, we refine the constraint, say, let's say, and this is just an example. So let's say we refined it to saying that we want now molecules which are less than or equal to 10, or -10. Now, obviously, guys who are less than or equal to -10, whenever that's the case, they're going to be less than or equal to -8 as well. And that's what that entailment sign is doing. So what happens is that that's on iteration I+1. So molecules that do not satisfy the constraint on iteration I, so bad guys, will not satisfy the constraint on iteration I+1. So, a molecule which didn't have affinity less than or equal to -8 on iteration I, they will continue to not be less than or equal to -10. So, but some of the good guys may turn out to be bad on the next iteration. So the context will change. So it's not the case that the objects play the same role in every context. So in context I, or iteration I, something might have been thought of as good and bad, and on iteration I+1, some of the good guys may become bad, so you will sample from a different distribution. Okay? So this changes the context of the prompt for the LLM and therefore the distribution. So this is what happens in the iterative process. So here are some sort of quantitative results. I'm just showing you a table on two very well-known benchmark problems for which the target site is known. We have existing results on from AI programs which have been used in the past to generate molecules, and these are well-known classic benchmark problems. One is called JAK2, and the other one is called DRD2. So JAK2 is the molecules is the left half of this table. The results for DRD2 are the right half of the table. So we have already known inhibitors, ground truth, which we know that these guys do bind to the target. Their mean, as you can see here, is 7.26, median 7.42. So it's a skewed distribution, obviously, but the range also is quite large. So of the range of known distributors is anywhere between 4 and say 8. Now this next one, VAEGNN, is the last, at that point, the best state of the art that we had access to. That was better than a number of other things that had been tried before, like deep reinforcement learning and so on. The VAEGNN obviously does what you would expect it to do. The VAE is the generator, the GNN is a discriminator. So the VAEGNN is basically like the LLM with logical feedback. It's basically an autoencoder with GNN feedback. So that one does somewhat okay. It produces molecules with somewhat lower mean and median, and seems to be even wider range of affinities when tested. So this GPT-LF plus, the plus part, I just assume that it's basically talking about some amount of initial background knowledge. So it's about whether we give it a sort of minimal background knowledge, or we give it all the kinds of background knowledge that we have as such. So plus is with the largest set of constraints. GPT is GPT-3, and PaLM was the best we could do at that time. Of course, now there are so many. You can see that the mean value for JAK2 is higher, medians are higher, certainly with GPT. Interestingly, the variation is also much lower, so you can get, and the same set of things are also happen with DRD2. So with a different target, you get higher affinities on average, and you get lower variation. Qualitatively, you can see this happening. So this is molecules on the left. This is for JAK2. Molecules on the left are on the first iteration. So you can see that on the first iteration, you get guys generated around 7 point something. And then on subsequent or later, this is the last iteration, things have moved substantially to the right, and mostly you're getting molecules generated. So the y-axis here is number of molecules. The x-axis is the binding affinity estimated by a standard. Where did that go? What's happened? Yeah. Okay. Same thing with DRD2. The first iteration, high affinity, but spread of them, and next iteration, things have moved substantially to the right, in other words. So this particular thing, I haven't shown the negative sign as such, so it's just easier to show it with just the positive sign. So everything is moved to the right, so it's actually over here, moving to the right is a good thing, as opposed to moving to the left with the usual negative sign of binding affinity. This is just to give you an example, a qualitative view of what is happening as the iteration proceeds. We did get the chemists involved to assess the molecules generated by the LLM. So the chemists were given 20 molecules of each for each target for JAK2 and DRD2, 10 for each generated by GPT and PaLM, but they weren't told who was which LLM was generating what. They assessed it on. I'm not going to read the assessment. There's a paper which appeared in this year's AAAI. You can read about their assessment over there. So they've assessed it on 2 dimensions: how potentially useful this would be as a molecule that would bind to the target, so on efficacy, and they identified some specific functional features, structural groups, functional, not in the mathematical sense, but in the sort of biological sense, or chemical sense, and using the patent literature, and identified the potential efficacy of these new molecules, and they also assessed them on novelty based on what they had already seen before. So on both fronts, I'm not going to tell you the exact details, but I can tell you that they were pretty happy on both fronts. They thought they were likely to be good binding molecules, and they were, not all of them, some proportion of them were novel. So that's for us a good thing. So now, so much for what we did. What do we plan to do now? We plan to take all this forward in 3 different ways. I told you that the results that I presented were all from known benchmarks, classic targets. We want to look at targets which we don't know much about. So we would like to look at targets where actually not much is known, and that's point number 1. And we're specifically focusing on cardiovascular disease. I'll tell you why in a moment. The second thing that we want, which we haven't done to any great extent, is that we haven't used any synthesis knowledge to any great extent. We've just used the knowledge which is there from the target site. So we haven't introduced constraints based on potential synthesizability other than a very basic one. So that's the second sort of thing. And the third thing is we want to also feedback. So we want to feedback constraints from synthesis. We want to feedback constraints from biological trials. So these are the three areas that we want to add to the logical feedback component. And we're doing this actually in a, so it's not all just sort of, hopeful pipe dream stuff. We're actually part of a big project which involves AI, which involves synthetic chemists, biologists, and also pharmaceutical industry to target specific cardiovascular disease in a kind of human AI interactive drug design approach. And I'm happy to say that we've made some progress already on number one, where we have managed to repeat basically whatever we did with the sort of benchmark data on the new target which was given to us for cardiovascular disease. And we've been able to generate new molecules, and we've also managed to make a little bit of a headway on number two, where once we generated new molecules, so we started with existing drugs which are in clinical trials at the moment for cardiovascular disease, and said, can you generate new molecules which have higher affinity than these ones? And we got a bunch of molecules. The synthetic chemist had a look at them and he said, yes, well, these are interesting, but I would like you to generate new molecules which have a particular substructure, which makes it easy to synthesize. And he gave us a particular substructure, and now we've added that as a special constraint in the logical component, which says that, so it's a predicate which says, does this have a substructure or not? If it doesn't, then it fails that constraint, and then it comes back as a bad molecule, and then we generate again. And now we are now getting molecules which are higher affinity potentially, because it's all simulated affinity. Testing has to come with some additional trials. So potentially higher affinity than the drugs, and also have the kind of substructure that the synthetic chemist wants. So we're making headway on 1 and 2. 3 will take some time, obviously. Turning to a bigger picture, this idea of LLMs with logical feedback is what I consider an instance of something which I've titled here as AI modular theory, or AMT. So AI modular theory, as it says at the bottom of the left bottom, is inspired by an area called SAT modular theory, which is looked at satisfiability in the context of existing theories provided as, say, background knowledge. So in AI modular theory, you come in with an initial theory, you update, this forms a prior, and then you update any existing prior that you have to give you a new theory that goes to a generative AI module, which is essentially consists of a generate and test. So it's a simple generate and test approach. Generates something. These T's over here are just transducers that change so that things can go into boxes in the right sort of format and syntax and so on. So they're just things that change the this input stream into an output stream. So the generator produces some output, which is translated by this T into some statements, which a reasoner can use, and the reasoner produces some inferences, which then the generator uses, and so on. This is done for a few iterations, and then out comes the result. The requirement that we want in all of this is that the reasoner is correct in some way, so the generator can be, quite strong, it can generate as crazy examples as it wants, instances as it wants, but if the reasoners are correct, that is fed back, and hopefully things will get corrected and the generation process will get streamlined into something more sensible. So by reasoners are correct, I mean that they should be, if you're looking at it as logic, as we were, they should be sound or calibrated, in other words, in a probabilistic sense. I'm not the first person to talk about this. Stephen Wolfram, as far as I know, talked about it for a long time, of linking up things like Mathematica to logical models. Rao and his colleagues have talked about it in the context of planning. So it's not a new thing, but it's an interesting way to use language models, I think. Even bigger picture that we're looking at in this area of cardiovascular disease is because now humans are involved. It's basically a kind of two-way intelligibility between humans and these AI modular theories. So humans send some messages and some predictions and explanations. Explanations can include background theory. Predictions are possible new labels on potential molecules. AI modular theory produces a new message with some new predictions, and this iterates. So I'm happy to elaborate on these things if people want, but there is a context in which we are developing all of these modules as such. So to summarize, I basically described what I think is a promising way to using LLMs to generate new leads for drug design. It uses two features of LLMs: their potential as vast repositories of general and specific chemical knowledge, and their ability to essentially generate or hallucinate new molecules given a fairly small numbers of specific examples as context. We use additional logic-based inference to enforce some sanity checks on the molecules based on chemical constraints on leads. We have not, although in the newer work we do, but we have not in this work that I presented used a key feature of LLMs, which is their ability to be instructed in a natural language and to produce explanations in natural language. So far, everything that I presented today was fairly constrained. We are doing this in future work when we have been involved with the chemist. Instructions from the synthetic chemist have been in the form of natural language, and we have incorporated that in that bigger two-way intelligibility setting. So I would like to stop by acknowledging a number of people and institutions that have helped put this work together. I was indeed class of 1981 chair when I was doing this work, and I was funded, the work was funded by AppCare to some partial extent. TCS Innovation Labs, TCS Life Sciences, and their research affiliate program were part of the funding that I received. Some of this work was discussed and developed when I was visiting on a sabbatical. I was visiting UNSW, School of Computer Science and Engineering, and the Center for Health Informatics at Macquarie. And I'm also indebted to the DBT for their project on developing predictive molecules, which is this particular project. So I'll stop there and take any questions. Thank you.
Are there any questions?
Seem to be 3 things on the chat. I don't know what.
No, sir. There's no questions from the participants.
Okay.
Sorry, I actually have a question that wasn't.
Yes.
So, I mean, so there is one simple contradiction which I'm unable to understand here, probably because I don't understand LLMs well. But technically, statistics is supposed to be drawing things from, the major part of the probability distribution. However, like most of the new knowledge would usually be on the tails.
Yeah.
So there seems to be some paradox or contradiction here, and I'm not sure how this is working out so well.
So I think the empirical results that suggest that these language models are few short learners is one kind of leg on which this is standing. So that one thing is that they have vast amounts of data which vast amounts of information that they've already compiled and ingested, which allows them, I think, to index into the correct sort of conditional distribution rather quickly. And whether that can be theoretically shown, I don't know how you do that, but at least in an empirical sense, it has been the case that if you do provide some few instances of the kinds of conditional distribution that you want to draw from, it seems to quickly focus in on those that part of the tail that you're interested in. How or why it does that, I don't know myself. I'm also surprised to find that it can do it so quickly without requiring too much data. Maybe it's some very very clever retrieval trick, which I don't completely understand. But that is one thing as to why it actually does it. The other thing is that the constraints themselves may be quite powerful in the sense that they may actually zero out large parts of the probability distribution. So if you sort of blank out large parts of it, then maybe it doesn't have much choice but to draw from the tail of the distribution that's left as such. So that's another possibility. It's not an entirely satisfactory answer. I should be able to give you an answer which says these are the
Okay.
This is the update rule. That's why it works. But I suspect these are 2 possible reasons why it's doing it so well.
Okay. So when doing these experiments, did you also try to change the temperature parameter for the generation, and how were the results? Like if temperature was 0, were the results better or worse?
I think we kept to the temperature of some 0.8 or something like that. The thing is, we want, so within all these iterative loops, there is one of the loops is that you sample several times from the LLM to get multiple examples in a batch, and then you give those to the constraint checker. So we want some variation in the examples, the molecules that are generated. We haven't done a rigorous study of systematically changing temperatures and seeing whether those with obviously zero temperature will sort of give us somewhat boring results, but increasing the temperature and whether that gives you more and more sort of interesting chemicals is something that is a possibility. We haven't checked that. Yeah.
Okay. Yeah. Thanks, Professor.
Neena, ma'am?
So, Professor Ashwin, how does this patenting work in this situation? Like, won't it be like somebody just takes over and patents every possible molecule that they can find? So could you just explain how does one regulate that?
Well, patenting is an expensive business. If you, each thing that you patent, but then pharmaceutical companies have a large amount of money, I suppose, and they can afford to patent a lot of molecules. But there's a big space, as I said, between 10 to the 60 and 10 to the 6. So, I don't think even pharmaceutical companies want to patent every possible molecule that can exist. And I don't think it's that straightforward of just saying that I want to patent this molecule. They have to show some actual reasons why you think this works and so on. So you can't just say I want to patent this molecule for cardiovascular as a potential drug for cardiovascular. But there are already the difficulty more for us is that when you generate molecules, the way it acts, this new molecule may look different, but it's still may not be patentable because the active components in that molecule, it may have additional things which make it look quite different, but the active components doing the actual job of lowering hypertension or whatever may actually already be done in some other by some other patented molecule. So that's a sort of interesting area that I've been thinking about how to incorporate that, whether you can sort of look at patent documents and say that is this molecule got something which is already been, or is it using some information which has already been patented? But so that's not the question that you asked. It's how do we stop people patenting everything? I'm not sure if you have the resources that you can't do that, but there may be some legal and some other checks which will prevent you from doing that. But for us, the more important question is how do you generate molecules that are patentable as opposed to if you want to do that sort of thing. I haven't been concerned with the patent side of things, but because we have a pharma company involved in this next stage of our work, I suspect that they're somewhat interested in that. But,
so basically, when you use an LLM which has been trained, we would assume that in some sense it will look at existing stuff which has already been patented, or that doesn't happen automatically?
Doesn't happen automatically because most of those it can look at patent documents that that exist in the public domain, but many of these, I mean, companies are developing LLMs to look at their own proprietary databases, which can look at their own proprietary databases, but we don't have access to those things. So when we are proposing these new molecules, we're only, that query, as I said, is just some known database, try to get things which are not in any known database, but it might be in some proprietary database and we don't know.
So, so the patent is reasonably for something showing effect of something, or can it also be for, you know, developing this molecule, in the sense building this molecule from, I don't know what, constituents or whatever?
I think
So-
for drugs, when you are patenting a particular molecule for something, you have to say what it's targeting and why that it is actually effective in doing something. I think you can't just say that. I mean, I'm not at all sure of what the constraints are, but I think you can't just say that. I think I'll patent this drug for this molecule for this problem. I think you have to show some evidence that it's actually useful for it, and that it doesn't infringe any existing patents. I mean, that's what lawyers will check to see that it doesn't infringe anything existing already. So one of the new molecules that we generated without the so I told you that for this cardiovascular problem, the second part, we incorporated some synthesis constraints. I don't know what the story is there, but one of the molecules that we did generate, which was the one which had some very good activity estimated from the simulation. All the biologists and the pharma guys said, "Oh yes, but this is not patentable because of something," and I don't remember what that thing was, but they said it wasn't patentable. So, and that then apparently stops it from being of interest to some lot of people anyway. But I mean, as a purely scientific thing, I don't see any bother to it. The fact that it may still be a good inhibitor is still true. And it may then form the basis of something which is patentable. So.
But it's a kind of chicken and egg situation, because you need to know that this particular thing will have this effect, which is already out in the open. And then you're trying to make something which will fit that.
Well, what is out in the open? So for this, what is out in the open is, say, in some cases, the actual lock and some keys. In the cardiovascular problem, the actual lock structure is not known, but something closely related to the lock structure is known. So we are using that as a kind of proxy. What is also known is some keys to the actual lock, which are being checked through on clinical trials and have had some biological tests. So we have some keys on the real lock. We have some sort of closely related lock, and we're trying to generate new keys to the actual lock using the proxy lock. So none of this is it the case that the AI is going to produce the final magical answer. You'll have to have people involved who synthesize it, who test. There will be tests on. So you do all of this, and then you'll find that it's either too toxic or it's not soluble, or all sorts of things may happen. Although you're proposing something new, it may not actually end up as a drug. But it's a start. So that's where we're operating, in providing the starts.
So typically, these companies would make many of these things internally, let's say.
Yeah, so they
And then
take one of these and then they will sort of produce
eliminate it.
variants of that with, so they have, chemical expertise, which will say, okay, we think that this is going to work because of these reasons, which is what they've already told us about some of the ones that we've generated. We think that this is plausible because it has these things, and we know those things do inhibit this kind of target. So then their job will be to produce some sufficiently, some variant of that which retains that essential feature, discards whatever else was found because they don't think that those have any role, and then try and test that.
So are companies doing this? Like, I mean,
Yes,
going
all
through
the time. Yeah.
just have been doing it.
Yeah.
Yeah. So we can't.
In fact, I have a friend at Merck who was saying just 2 to 3 days ago that it's almost nauseating how many internal meetings he's been in where people are talking about the use of AI to do something or the other in their drug design discovery process. So companies are doing this for sure. So
So this would be a new line for them in the sense.
yes,
Instead of just the chemical pathway, they will look at the AI pathway of drug
yes.
design.
So they just use the AI. So they use the chemists to produce something, and then say,
Right.
here's something. Can you find something better than this? And then they'll test that as well. So that sort of thing is going to happen all the time.
So basically, these drug companies would now have their own set of AI-based tools.
Yes,
which they have
absolutely,
set up internally.
Yes, absolutely.
Good.
I mean, they've always done it like that. I mean, a sort of case in point where I remember a colleague of mine gave a talk somewhat like this, and to a drug company, and then they were in talks on using his technique to develop something. This was a long time ago, so it's probably not a relevant technique. But by the time he came back home, they told him that we've already implemented this based on the paper that you wrote, so we don't really need to talk to you about it. So
Okay.
yes, so that part of it is always there. That
I mean, we put it in the open, and they would be like,
yes, it's I think,
taking it as knowledge and using it for their.
well,
and we would
I'm not saying that they're all sitting around skulking and doing this in some covert way, but I'm saying that it's sort of part of the bigger thing of running a big pharmaceutical company, is that you have to be aware of current developments and then see how it fits into whatever you have. So, I mean, that is their reason for their existence. So they have to do it that way.
Now they will be looking at AI part of it, in the sense they would have to hire experts who can look at the AI knowledge which is coming and saying, okay, this might be useful for us. Like not
Possibly,
just the chemical.
possibly, but the one thing that has happened, as you know, is that the almost all code, like all this code, is all in the open domain, so they can take whatever. So they don't need that much AI expertise as such. They just need programming expertise and such, I suppose. But yeah.
Thank you so much.
Yeah, thank you. Any other questions? Anybody else?
I don't think so, sir.
Okay,
Yeah.
right. Well,
Sir, thank you, sir.
Thank you, Srinivas, for running this talk.